I'm going to mute myself now.
All right. So tell me when to start, and I will start showing. Okay.
Sure. I'm waiting for Shovan for joining.
Yeah. You're going to get
Yeah,
introduced
Shovan called me
instead.
up some time ago.
Okay. Yeah. So your volume and everything is good. Perfect. So I think
Good.
we should be good. So I'll give a brief introduction. Then you can start after that.
Okay. All right. Sounds good.
Okay. So hello everyone. Am I audible? I think.
Yes, Umik.
Okay. Thank you. So hello everyone. Welcome to the Department of Chemistry, IIT Delhi. And thank you very much for joining this webinar today. We will probably give a few more minutes, like a couple more minutes for the participants to come in. Okay, probably we'll start. Uh, so my name is Soumik Siddhanta, and I am the webinar facilitator and an assistant professor at the Department of Chemistry, IIT Delhi. And we are very pleased to bring you this content today, which is the part of online Pratidhwani series that the department is organizing. Under Pratidhwani, we invite eminent scientists from the different areas of chemistry from all around the world. These 30-minute talks are held every Thursday afternoon. Please do check out our webinar website. You can find the link in our flyer emails and also on our department website. The webinar series is organized by Author Cafe. Author Cafe is an online platform that aids with writing, collecting, organizing, collaborating, and publishing your research content through features and integrations with products like Mendeley, Zotero, Orchid, Crossref, and so on. Think of it like Google Docs, specially designed and developed for the academic community, and much more. Today, we are thrilled to have Professor Sudipta Maiti from Tata Institute of Fundamental Research, Mumbai, as our distinguished speaker. Following Professor Maiti's talk, Professor Shobhan Sen, Associate Professor at the School of Physical Sciences, Jawaharlal Nehru University, New Delhi, will present a short summary of the talk and also take a few question and answers. Before we start, a couple of technical details. Please do not unmute yourself during the talk. If you have any questions, please type them in the chat box and we will try to get at least some of them through to the speaker after the talk. Professor Maiti graduated from Indian Institute of Technology, Kanpur in India with a degree in physics. He then joined Bob Knox at the University of Rochester for postgraduate studies. He obtained his PhD from the University of Pennsylvania in 1994, working with Robin Hochstrasser and Les Dutton. He worked on femtosecond vibrational spectroscopy of myoglobins and the photosynthetic reaction center, and this is where he learned the art of biophysical laser spectroscopy. He then joined Watt Webb at Cornell University for a postdoc and worked on protein kinetics using fluorescence correlation spectroscopy and also on 3-photon imaging of neurotransmitters. He returned to India with a faculty position at the Tata Institute of Fundamental Research, Mumbai in 1998, where he is currently a professor. He is interested in the problems of protein aggregation and folding, peptide membrane interactions, monoamine neurotransmitter dynamics, among others. He is also involved in the development of various fluorescence spectroscopy, imaging, and related biophysical techniques. He has mentored several PhD students and postdocs and have received several awards, including the Wellcome Trust Senior Overseas Research Fellowship. A very warm welcome, and over to you, Professor Maiti.
Thank you very much, Shomik, and thanks to Shomik and Professor Ram Ramaswamy for this warm invitation to give this lecture. It's great to be in IIT Delhi. I know that's a sad joke in this COVID times. One of the few things that are good about COVID is the marvelous sunsets for 6 months every day in a row, except for the rainy days that you have had from our little private seashore that TIFR has in the southern tip of Mumbai. So here you see the sun taking a dip into the sea. And actually, for the rest of the talk, I'll talk about what happens when proteins take a dip in the water. Okay. So before I start, let me just introduce what my lab does, because I'll talk about only a little small sliver of what my lab does. So we are interested in how proteins aggregate, and proteins which do not have a structure to start with, when they aggregate, how do they get a structure? So how do they get a structure in solution? How do they get a structure in the membrane? And we keep inventing techniques. That's something that we have a lot of fun with, to do these things, because these are not problems that you can solve otherwise very easily. And then we are also interested in knowing when these structures form, which are called amyloid structures, which are associated with different diseases such as Alzheimer's, Parkinson's, etc. Why are they toxic at all? Okay, these are peptides, proteins, they shouldn't be toxic. Why are they toxic at all? And then we also look at other things. Something has been my passion for more than 20 years is multiphoton imaging of neurotransmitters, trying to understand how messages get passed on between two neurons. This context, we're having a lot of fun this year and last 1 year or so, knowing and understanding something that we never thought about. Nobody ever thought about, that the small molecules called neurotransmitters, serotonin, dopamine, these are molecules which pass on messages between the brain cells, how they interact with the membrane. And most likely, that's what we found in the last several months, most likely there is a parallel pathway for signaling that is independent of a receptor that this serotonin or dopamine can attack, which is something that nobody knew about. And we are very excited, and a lot of work is going on in the lab to understand this. We, of course, also develop a lot of spectroscopic tools, high-end spectroscopic tools, including single molecule tools, including some that have been commercialized by companies in India. Okay, so let me just get to the stuff that I want to ask today. Proteins are rather big molecules. Okay, how big are they? Well, if you talk to a synthetic chemist, again, COVID will keep coming back into this talk. Indeed, that's something that is engrossing all of us. This is something, hydroxychloroquine, showed a lot of promise in the initial things, or thought to be promising in the initial stages, which unfortunately didn't come out to be true. This is if you talk to a synthetic chemist, he or she would say it's probably a mid-sized molecule, not small at all. However, if you put it next to a protein of the virus that it is supposed to stop, this is the spike protein. This is a protein that is like an X, opens the door of a human cell. It is tiny. You can see this little red boundary. This is 330 Daltons, and this thing is about 500 times bigger. As by no means it's the biggest protein known. Proteins are known, megadalton proteins are known. But you realize that we typically make molecules which are of the order of 100 Daltons. Nature makes molecules which are of the order of 100 kilodaltons. That's standard stuff for biology. Yet, they're both designed to do some chemistry or some interactions. Okay, why is it that nature makes it that big? And the question is relevant because if you look at how nature does chemistry, for example, looking at an enzyme called trypsin, which snips protein molecules at particular places, you see the business end of the protein. This is the whole protein, okay, stylized in terms of ribbons and all. This is about a 23.3 kilodalton protein. The business end of the protein, where all that chemistry goes on, is this little bit. So it doesn't use the rest of it. There's a zoomed up version. A serine, a histidine, and an aspartate actually make the bulk of what is the business end, what does chemistry. Yet it has this huge, and this together would be about 0.3, 0.4 kilodaltons, and which is roughly 1% of the mass of the whole protein. The whole rest of the protein does nothing useful as far as we can tell. Why do they have to be so big? Okay. So there is another example. As I said, they'll keep coming back to COVID. Catalysts like enzymes do chemistry, but there is a lot of biology also happens when two molecules come together. In fact, that's sort of the beginning act of biology when two large biomolecules bind to each other. So here is the human ACE2 protein, which sits in our membrane. And here is a small part of the called the RBD of the COVID, the virus that that is responsible for COVID, SARS-CoV-2, spike protein. Okay, the spike protein again is big, but this is how they interact. This is how the X is opening the door to your cells, okay, to our cells. And if you look at it, that protein interaction almost wholly is mediated by this one single alpha helix that belongs to this ACE2 protein. Okay, that red one marked here. That's 23 residues. Nothing compared to the rest of the whole huge protein complex. So if I just snip out these 23 residues, will it work? It's not only a fundamental curiosity that the nature is making proteins so big, yet only a few of the residues are actually at work, and can we replace them by those few residues, but also a very practical question, because if we can, then we can easily make small molecules, small peptides, not small molecules like this, flood the system of a COVID patient and hope that the binding sites of the spike protein all get saturated by it, so it doesn't bind to a cell anymore. In fact, that's still, even now, is one of the strategies people are trying to follow, who are trying to come up with peptide-based drug design. We also wanted to make an indicator for this by labeling it fluorescently. And one of the promising pathways was looking at this. Well, I've been dealing with proteins for 30 years, longer than some of your ages. So I knew right away that if you snip this part open, it won't stay like that. Okay. Still, there was some hope from some reports which right now has turned out to be false, that there was supposed to be some nanomolar binding of this protein to this peptide, just the 23 residues to this. But actually that was not true, as has been proven lately. And let's talk about whether it works and why it doesn't work. So one of my students, Anirban, he was tasked to make this peptide. He's very swift with these things. He made a 23 residue peptide. And we looked at the CD structure, CD of the structure. CD is circular dichroism. Here is a spectrum. And you can see that for those who have looked at a CD spectrum of a protein and you're expecting an alpha helix, you'd expect something at 208 and 222. It looks nothing like an alpha helix. If you take some program and analyze it, maybe 2%, 3%, and we're talking about, our hope was 90% would be alpha helix. So it doesn't work. It's exactly that part, which is nicely alpha helical in the main protein, supposed to interact with the spike protein. You snip it out and it doesn't stay as an alpha helix. Neither, and you have tested it with spike proteins, neither does it attach to the spike protein. Okay, bummer, but expected. Why? Because there is something called folding. The rest of the protein is holding these residues, which are at the business end, at a particular geometry. There's, of course, dynamics, etc., are also there. The first of all, it has to hold it in a particular geometry. And the protein can hold, the protein is a rather floppy molecule if it's unfolded, can hold things in a particular structure if a lot of it is together, because it spontaneously folds due to various interactions, but one of the major ones is called the hydrophobic interaction. Okay, you can roughly get two color code for proteins, most of the protein. One is blue, which is hydrophilic residues. Protein is a long chain with residues hanging from that chain, and hydrophobic residues, which are marked in yellow here. And you see how a protein such as myoglobin folds. The myoglobin actually tries to wrap itself up with sheets of hydrophilic residues, which will face the water, because most of aqueous proteins are encountering water on the other side, and hiding all its hydrophobic residues inside. And that's one of the cardinal principles how proteins fold. Okay, and if you take a cut through the middle of this protein, you will see much of it is inside is yellow. So there is a cut through the middle of myoglobin, and you see a lot of these yellow things. So it's all hiding its yellow yolk, you call it, the hydrophobic residues, by wrapping itself up by hydrophilic residues. Nice, simple organization. In fact, you can go far trying to predict how and why protein molecules will fold just with a two-color code. Of course, there's much more to it, but this is one of the driving principles. Okay, so what happens when you take a snip a little peptide, does not have enough material to cover itself with hydrophilic residues. Not enough hydrophilic residues, not enough hydrophobic residues which can form the core. So this kind of core shell architecture is impossible to achieve when you have very few residues. So there are very few examples of proteins done just right. A 37 residue protein does fold, etc. But by far, it is the expectation that if you take a very small protein, and they're called peptides, if they roughly you have less than 50 residues, they're called peptides. Peptides won't fold properly. Okay, can design very well to fold them, but most peptides, especially snipped out from a protein, won't fold. The rest of the protein is there to hold it in that form, to exactly have the key, hold the key in the right form so that it can open the door. Okay, so what does it mean? Well, there are ways that a small peptide can achieve a folded form. In fact, it's done in nature. Okay, so this one of the ways, and there are three ways that I'll talk about. Two of the ways are here. The third one is the meat of the talk that will come towards come at the end or in the next half. This you see is a few molecules. Okay, red and green. There are few molecules. They're each short peptides. The short peptides can find each other. So any one of them doesn't have enough of the material, hydrophobic to form the core and hydrophilic to wrap themselves up. But if 4, 3, 5 of them come together, there may be enough material available. And that's exactly what nature does. Okay, and you can have these peptides fold together, co-fold together in a form which is at least partially folded, some unfolded parts, and it can take up a structure. not probably the structure that it had when you snipped it from the original protein, but some structure. And this is kosher folding. The folding does happen. The other option for it is to find a place in the cell, and in a living cell, where it doesn't have to hide the hydrophobic parts. So you can go to a cell membrane. These are lipid bilayer. So this is, you can see an alpha helix nicely inserting itself in the cell membrane. It's probably very hydrophobic on the outside. Doesn't have to hide it because it went away from water. And we see frequently that both of these happen for many of these peptides, which have many hydrophobic and some hydrophilic residues. They do get together to form small, what we call oligomers. And then even after the forming, there is enough of hydrophobic residues. They are somewhat soluble in water, but much of them like to go and insert into the membrane. In fact, that creates problems because, as I said, nature does, for reasons that we don't understand very well, create many of these peptides. One of the most famous ones is amyloid beta. So this amyloid beta is a 40-odd residues peptide snipped from a bigger protein, much bigger protein called amyloid precursor protein, which sits in our membrane, your membrane and mine. And amyloid beta, for some reason, maybe it's an accident of evolution or whatever, there are things that snip it. And in some cases, they will come together, form an oligomer, and those oligomers are toxic. They're so toxic that they start killing brain cells. It mostly happens in the brain, and people get Alzheimer's disease, which is not an uncommon disease at all. People who are above 80, about 40% of them have Alzheimer's disease. And much younger people can also have it. So one of those big unsolved mysteries of why it is happening, how it is happening. And it's just one of the family. There are neurodegenerative diseases of many kinds, but they all come back to this fact that things that don't fold properly by themselves as a monomer can fold as an oligomer and somehow create toxicity. And it's very hard to get their structures because, aggregation-prone proteins, if you put them in high concentration, which is required if you want to look at the structure by some means such as such as NMR or X-ray crystallography or something, you need a lot of it and a lot of high concentration. You can do it. You can do it. So we spent years thinking about ways to do it. Finally, cracked the problem open to a large extent using a combination of single molecule techniques, which are very sensitive, can go down to nanomolar, picomolar level, and solid-state NMR, and doing something, flash freezing, etc. It's not the story today, but we finally were able to understand some of the structural parameters of how amyloid beta oligomers create toxic assemblies. That was not all. Amyloid beta oligomer actually has a large tendency to sit in the membrane. And if you talk to the community, I would say 8, 9 out of 10 people would say this is how it sits in the membrane, should be the key to understanding why amyloid beta is toxic and causes Alzheimer's disease. Same story with another peptide, similar length, called amylin, which causes type 2 diabetes. And there are many others, the Huntington protein, etc. So we again invented some methods and looked at it to try to understand how these peptides, which by themselves cannot fold, are taking up structure, and the structures are sitting in the membrane. Membrane is a very different environment. The structure can change and probably does change to some extent, again using new methods based on surface enhanced Raman spectroscopy, etc. We could get to something that does something about the structure of this protein. So again, that's not a story I'm going to go into. We didn't know till very recently, till earlier this year, how many of them are going together, because SIRS told us what kind of secondary structure that the peptide has. So we had to depend on the person who graduated 2 weeks ago from the lab, Shimli De, and it's her ingenuity that showed us a way using single molecule photobleaching technique. You go look at one by one these assemblies and count literally how many particles are there in that assembly. And that told us that it's basically, for amylin, it's at least trimers and tetramers. Okay, come back and see that whether these sort of structural understandings get us anywhere. And here is the start of our fun part. So we knew that amyloid beta in the initial part has a very crucial connection between two residues, one phenylalanine in the 19th position and another leucine in the 34th position. And we said, okay, they interact each other. It's a hydrophobic interaction. Now, can I fool this intramolecular interaction with something, some small peptide, which will let F19 and L34 think that they're interacting, but yet they're not interacting. So we came up with a peptide FRFR, phenylalanine, arginine, phenylalanine, arginine. Okay, the phenylalanine, if you put this peptide in the solution at a reasonable concentration, the phenylalanine, the logic was very, very simplistic. In fact, the phenylalanine was expected to interact with L34, lulling it into believing that it's interacting with F19 because it can't tell F19 for another phenylalanine. And of course, we could put LFL, and then this phenylalanine will think it is interacting with the leucine, but that would have been an insoluble peptide. And so we put in a charged residue, arginine, because we knew that phenylalanines love arginines, which is slightly counterintuitive, but if you look at the quantum chemistry of it, there's a cation pi interaction between the phenylalanine and the charge of the arginine that makes this a rather favorable interactions, about 2 kTs of energy. So FRFR, and we created some related things, but this is the first decoy that we made in the lab about 3 years ago, trying to fool nature into believing short peptide is part of something else. And it worked. So as I said, the oligomer becomes toxic by binding to lipid bilayers. So we can measure vesicle binding as a quick probe of whether this is going to work or not. And if we put FRFR, just the peptide, with amyloid beta, you see the vesicle binding drops by almost a factor of 10. Okay, and something else, A beta phenylalanine or alanine or arginine itself, or even FK doesn't drop it by that much. So this is our beginning of a journey. Just think of the structure, try to replace that interface with something that looks similar, but it's not similar. It's not the same. Of course, we had one great advantage. It's FRFR, 4 peptides never folds. So we didn't have to bother about folding it. Just dump enough of it and it should work. But if I think about a bigger helix, such as that helix of the spike protein, the spike binding peptide, the helix belongs to the S2 protein, the spike binding peptide, then we have to somehow keep it stable. How do you keep an alpha helix stable? If you look at the left part, this is an alpha helix that some sculptor built in front of the house of Linus Pauling in Portland, Oregon, USA. This is where Linus Pauling, who discovered the alpha helix structure of the secondary element structure, alpha helix, and this is where he was born. But you see, if this was made of proteins like noodle-like objects, this would collapse. This is made of steel. It's something that gives a structural rigidity to that stylized alpha helix. So if we take a real alpha helix, which is here, the picture of one, we argued, how about you do some chemistry? Okay, we don't have to be nature. We can play little more intelligent, perhaps compared to nature, and we know the chemistry. We'll put staples. How we use staple pins, which can join 2 things together. We put staples, hard staples, and maybe we'll be able to stabilize structural elements. Okay, this is what we are working on right now. But between these two, that hopefully another time I'll talk to you about stapling an alpha helix. The initial results are coming in. But I'll tell you something about a very exciting thing that we first started with when the stapling idea came. We asked, okay, can we fool? So this is one part of the protein, right? I staple that part. Hope that that part actually is retaining the secondary structure that is supposed to retain and present it to that protein itself. Would it exchange it to its own own part of it? You realize, there's a here and protein has a hand, and suddenly he sees another hand, and the false hand, which is a decoy, comes in and sits in the place where the hand is, because it interacts with the rest of the protein. Should be exactly the same way. And especially when the protein is trying to fold, it's not yet stable. So we looked at proteins. Protein folding is a fascinating subject. Some of the proteins, small globular proteins, fold through nucleation. So first one of the part folds, and the rest of the protein then folds around it very fast. So this protein, ubiquitin, a small 76 residue protein, has a nucleation pathway. It's one of the major victories of computational biophysics showed, D.E. Shaw, using his Anton computer, showed how ubiquitin folds. And it folds by forming this part. This is the whole ubiquitin first, and the rest of the protein protein folds around it. We decided we'll make this, and we did. And when you make it, it doesn't remain folded. But when you put a staple, it gets barely folded, okay? Or it's closer to a folded state than unstapled. So this is our chemical staple, okay? It's just two cysteine residues stapled together by a disulfide linkage. And we presented it to the unfolded protein, asked, can we fool this protein into folding or folding faster, okay? And this is what we found. This is what, again, Anirban's work, Anirban Das, what he found. This is the kinetics of red, is the kinetics of folding. Without this peptide, which you call the xenonucleus, sorry, blue is the kinetics of folding. Without the xenonucleus, red is with the xenonucleus. It's not that much of an effect because its own peptide is hanging around, but it is an effect that is very clearly discernible. You see here, ubiquitin without and with the staple peptide. And a different final, so you drop it from guanidine, high guanidine to low guanidine, and look at it, and you see every time the folding becomes faster by a factor of 30, 35, 40%. And it is not a random effect because it is clearly dose dependent on the decoy peptide that we are putting in as the nucleus. So we're presenting the nucleus, so the rest of the protein folds around it. A big question is what happens to its own nucleus part, and that's something that I'm not going to go into. I don't have time. Shomik, how much time do you have? Shomik?
Yes, so probably like 5 minutes or
5 minutes. Okay. Yeah, I'll cut out those parts and we'll not talk about it. And it's also sequence dependent. So you can take the same one and you can garble the sequence up. It has some sort of an effect due to just the generic nature of the peptide, you see, but the effect is much less. So you can present the protein, which is unfolded, and it's a kinetic experiment, just trying to fold as you drop it from high guanidine to low guanidine buffer, and you present it with the staple peptide, it folds much faster. Present it with the same peptide, but the sequence is garbled, stapled random, and it does fold a little faster than when it is without a peptide, but much less than when it is in the proper sequence. And finally, Ranabir Das did this experiment with us with NMR. It showed that yes, there is a weak interaction that can show clearly where this decoy peptide is going is the same place where the normal peptide would be sitting. So there's a proof of principle where we have made a decoy which induces folding into a protein. So imagine we just picked up the nucleus, all small, or let's say majority of small globular proteins, you might be able to find a nucleus. I'm getting a request for annotation. Okay, I don't know about that. Sorry, Kunal. So I'll talk to you later about it. So there's a generic principle how you can modulate the folding of a protein by intelligently presenting chemically stabilized nuclei. So you look at the protein, suppose you find out by experiments or by molecular dynamic simulations what the nucleus is, and you make a lot of that nucleus, put it in that, and it should affect the folding only of that protein, even if it's with 1,000 other proteins. So it's a protein-specific folding modifier. We are pretty excited about it because it can have applications. A peptide can, in principle, be expressed in a cell, etc. And ubiquitin is a very important peptide. But we will be really looking at other proteins. Ubiquitin is a very important protein, does a lot of stuff. We're looking at other proteins and see if we can specifically modify the folding of things by short peptides. So this can be done. Let me try to wrap up by telling you what I have told you and where we're going. Okay. So where are we? What have I told you? So first I told you that the regions of a protein that direct the binding or chemistry that is done by the protein are small compared to the protein. So why does the rest of the protein exist? Well, if we synthesize these regions independently, they would not do the same job. Okay, they're supposed to, but they would not do the same job because they would not be able to fold. So it does happen in nature. You do have snippets of actual protein through some enzymatic cleavage, etc. And that is a whole different ballgame because they do fold by aggregating, trying to hide its hydrophobic residues, etc. They attack cells, and a lot of the diseases we cannot solve are due to these things. But also, if we think about the palette of chemistry, adding the palette of synthetic chemistry to it, we can reproduce the original structure by shaving off the rest of the protein. That's really the dream. You preserve the active site or preserve the interface, biological interface by which two macromolecules talk to each other, attach to each other, with just small molecules held exactly the same way. A lot of chemistry needs to be done. What I showed you are just little examples of this, but this is an exciting area, and some people have already started doing it. For example, one of my young friends, Ishu Saraugi in IISER Bhopal, she works on helix stabilizers, artificial molecules which stabilizes helices and all, and we were working with her. And there are other people who are thinking about it, but this is a very fascinating field because a lot of your biophysical intuition has to come in. The synthetic chemist has to walk in, the molecular dynamic simulation person has to walk in, and the biologist has to be there. So, and we can change how a cell works by putting in the right kind of peptides at the right time. So, a short peptide I showed you can disrupt interactions of amyloid beta or can modulate folding rates in ubiquitin. Okay, so it's a new palette of mini proteins that can mimic much of what the large proteins do. Do away with the large genome, just have little things. Is that what we are coming to? So my last slide. Yes, why not? Okay, so we can design a whole world of mini proteins. And I do not have a slide for this, but if you look at this challenge of making the helix stabilize for the spike binding peptide, just 2 weeks ago, there is a science paper which showed a minimal protein, which is just 3 helices, taking the rest of this, that exactly holds it like nature holds it. That they are already peddling as, proposing as a drug candidate. Okay, so we can have a whole world of mini proteins, should be very interesting as pharmacological agents, but also we can, for fundamental biology, we can change the interaction of specific proteins inside a cell, and we can have bioorthogonal chemistry. So you can supply something from the outside of a cell. It should be cell permeable, maybe flash a little UV light or something, and create those staples at the right places if you create the proteins in the right way. And you can reprogram cells at will with these little things that nature doesn't know about, but does some of the things that nature took 4 billion years or 3 billion years of evolution to come with. So maybe you can create a new artificial living cell or a hybrid cell where some of the proteins are nature's and some of the other little things does exactly what the other proteins do. There's this big race of taking the minimalist living cell, making it artificially in the lab, 322 genes, some people say. But perhaps if you replace some of those proteins by these little peptides, it can come down to a small level. So it's a lot of fun, and we want to replace proteins for practical and completely useless purposes, but for fun purposes with their Lilliput counterparts. Okay, so I end my talk here by thanking the people who actually are doing all the hard work, and well, I'm having fun calling them up and saying, hey, why don't we try this? And then they work hard for 3 months and come back saying, oh, but you didn't think of that. And I have to hang my head in shame and say, yes, I did. Sometimes they show the way, but look, we thought of that. So 2 of the people whose work I have talked about here is Bappa, who has graduated and has gone across the seas for a postdoc, and Anirban, who is just beginning to write up his thesis right now, to very gifted, talented students. We are really blessed with talented students in TIFR, and Anirban did all the work, is still doing the work on that xenonucleus peptide. Lots of students I cannot name, too little time, and lots of collaborators from all corners of the world, including from TIFR, who have worked with us at different times and are still working and are excited about the stuff that we are doing. Okay, and financial support comes from DAE, because TIFR is a DAE institute, and also from DBT. Thank you very much for listening to me last half an hour.
Yeah, thank you, Professor. for the insightful and stimulating talk, and also like for addressing a very important question that comes to all our minds who have studied about proteins and their functions. And now we are going to have a short discussion session, which will be followed by another Q&A session. So I request everyone to type out your question in the chat box if possible. And for this session, we have Professor Shobhan Sen from Jawaharlal Nehru University, New Delhi, who will take over this discussion. So over to you, Professor Sen.
Okay.
So I will probably make you the presenter now. So this presentation will go off, but then we can put it on later during the Q&A session.
Should I unshare or something or stop sharing?
Not necessary. I'll just,
Right.
yeah, so it will go off. Anyways.
Yeah. So yeah, thanks, Somik. I think now it's working to some extent because initially I had some issues with the video, so I'm now using my mobile phone. Somehow it's working now. So it's anyway. So thanks to Professor Maiti. In fact, Sudipta. We always call him Sudipta. So that's the fascinating always to listen to his talk from a stalwart and from a teacher, I would say, more of a teacher that he can really talk about something which is so much in the forefront nowadays with so many new ideas, to be very frank, that these ideas of coming to the small parts of the protein and how can you really tweak those small parts to change its nucleation properties and the other things. That is something very, very interesting to really know about, because obviously I have heard so many of your talks, but this is probably the first time I'm listening to this part of the talk, that you already tried this kind of staple part which can really change the protein structure altogether, and you can control the structure. So I think if I really try to in a sense summarize for the students as such, obviously everything has been told by Sudipta, but the very important fact that he mentioned, that if you really go down to this molecular level, I think the main part that you have to really tweak and to understand the tweaking, what to be tweaked, is that the hydrophobic interactions or the interfaces, the interactions that is really happening at the interfaces altogether. And I think that's exactly been nicely pointed out by Sudipta here, that if you really know from, let's say, molecular dynamic simulations, like David Shaw and everybody has done, say, big simulations with the Anton machines, and if you know those, all the conformational spaces, and automatically you know these nucleation points, and if you can now try to tweak those points to really try to do something with a smaller, much smaller part of the protein. Because, Sudipta has already given several of the examples, and I still remember during my postdocs, I was working with one of such kind of a system like AP endonuclease with the DNA, and how this AP endonuclease really comes and try to find out this abasic sites where you have a damaged DNA. And it's very fascinating to see that only one particular aspartate really comes and try to find out that region to cut this particular portion of this abasic site. So that's absolutely fascinating, because if you really look at that AP endonuclease protein, it's so big, but the actual enzymatic process really happening in a very small part. The very similar example, which is to some extent, meaning we're trying to also understand in many ways that how the mismatches can be, this targeted by this different mutase H kind of protein like this. But again, you see those places where you have a very small part, which is really responsible for all the enzymatic processes really happening. And that's exactly what has been given the examples for several of these ideas into the proteins that Professor Maiti has really looked into. And I believe that we will be able to really see several of these kind of fascinating ideas in the many other areas, which Sudipta already has mentioned, that several groups in ISA Gopal already trying several of these things. And so this really makes a very big challenge to, in a sense, challenge the nature, that how nature really works, and can we really try to mimic some part to make something going altogether. So I believe the research done by Sudipta is probably really towards that end in a big way. So we are really hoping to have more interesting ideas on this particular line. And we are learning all the time. I think this is the first time I'm listening to you this talk, and I've never heard about this part of the talk. So maybe I can also have some more opinion on this on Sudipta, from Sudipta, in a sense, if you want to say add some more things in the discussion. In the which direction really we want to go ahead, meaning do we really want to tweak the nature in a big way or rather a small way for some industrial applications or whatsoever? What is the way forward?
I think the goal is it's a twin goal. One is something that is applied, like people are trying to find out how to fool the SARS-CoV-2 virus. That's, of course, down the pharmaceutical space. But it's also the ability to tweak particular proteins inside the cell with some small peptides, which you may be able to make it cell permeable at defined time and space. And it goes in and disrupts only that protein. See, it's very difficult to target a particular protein without coming up with an antibody or something. And then you are talking about a dead cell. So if, on the other hand, the cells, the proteins inside the cell are not absolutely stable. They're partially opening up all the time. So if you know where they're not so stable and you can put take a part of that, make it very stable chemically, supply it to the cell, probably it will go and attach there. So it's a way of targeting particular proteins by mimicking part of the protein itself. I think that has a lot of. So we always think of antibodies, large things that will recognize things. How about small things which will have recognition potential? How about having a general principle that can be computed to make those things? Look at the molecular dynamics evolution and says, yes, if I make that and somehow I'm able to stabilize that, either a helix or a beta sheet or something, and I put it in, if it's around, the protein is opening up occasionally in that part, it'll go and enter and disable the protein or do something, whatever. It will be fun. And most proteins that you simulate, you will be able to guess such parts. So it's really more fun than for the pharmaceutical part that I'm targeting things.
Yeah. So I think that's really nicely explained in that way. So as in fact, you all were mentioning also in the last slide when we were concluding that already these small peptides have also been used as a kind of a drug in many ways nowadays. Like small peptide drugs can be also used for targeting a particular specific target in our human body as such. So these ideas is already been there, and probably in a great application is there. But I believe from the academic point of view, if we really try to see this, how the protein structures really get stabilized, even if you tweak a bigger part, keeping the small part of the system, I think that's a new understanding or new ideas that you are really putting into in this particular work that you are doing in your lab, I think. So I think this is a fascinating field to explore, and as he already said, that we need several people around, that chemists, the biologists, the computational people, and I think that's the way we can probably go ahead and think about much bigger story about these protein foldings and how we can tweak those things, I believe. So anyway, so I think we don't have sufficient big time to really discuss too many things. I think we have to anyway go to the Q&A sessions. So I think I will anyway hand over to Somik, because Somik is looking at all the questions right now. So if we have some more questions anyway, we'll discuss those questions, and new things will come up anyway. Yeah, please, Somik, go ahead.
Yeah, thank you, Professor Sen, for nicely summarizing and holding this discussion session. So the floor is now open for questions. So if anybody has any question, either you can raise your hand now, or I can already see a few questions in the chat box. Okay, and also, interestingly, our this webinar is being streamed live on YouTube right now, and there are a couple of questions there too in the chat box. So probably I will take them, one of them. So the first question is from Tushar Ranjan Panda, so who wants to know what kind of staples that you use as stabilizers, and whether these staples are site selective in biological sense? So this is the question.
Very good question. So the short answer is no, they are not very site selective yet. The staples that you're using right now are actually extending the peptides to just have 2 extra cysteines and somehow oxidizing those cysteines to have a disulfide linkage. The other staple that we are working with for stabilizing an alpha helix is a staple which has a benzyl ring and then exact distance that can also do some thiol chemistry between 2 cysteines and have the exact distance that's the turn of the alpha helix. And the idea is that if we staple few of the turns, then the nucleation of the alpha helix is actually difficult. Once it nucleates, the rest of it can come together, and those chemical staples are available. There are several other staples. I mean, there is a trimeric staple that somebody has made, which holds 3 helix bundles together. So basically, it has to be done with the protein in mind. If you have the protein in mind, if it's just a generic alpha helix, then there are generic solutions. As long as you can put a couple of cysteines or other reactive residues somewhere. But if it has to be some larger parts of the protein, a 3 helix bundle or something, you have to think of the chemical space. So that's why I said a synthetic chemist has to be your partner if you're a biophysicist in this, and you're a computational chemist. So you can think that the synthetic chemists have a large palette of at least not so large molecules, but molecules which are large enough that can hold things together. Ultimately, is the entropy. The protein wants to unfold because why should all these particles be in a given space with respect to each other? And if you put a few constant at the right places, then that's all it really requires. The rest of the protein has no choice but find the energy minimum and fold into the way that the protein is. So you have to look at the interaction and come up with it. But the short answer is we do not have really protein-specific ligands. What we do is that we can artificially create these and put our cysteines at the right places so that we do some chemistry, it happens right there.
Okay. So, so the next question is from Suru. So he asked that how nature controls the selective synthesis of alpha helix and beta pleated sheet structures in metalloenzymes.
Okay, so I mean the alpha helix and beta pleated sheet structures are not exclusive to metalloenzymes. They are there everywhere, and the question is, how did nature decide what sequence to put in where these things will go? So again, the answer is that yes, if you look at the residue space in a very simplistic way, you can give a score of residues which are prone to be in an alpha helix, given their 3-dimensional structure, other residues which can form better sheets, and you can give scores. But of course, it's never just those residues. Otherwise, I can just give you a formula of residues and say, oh, this is going to be an alpha helix, or this is going to be a beta sheet. Frequently, it's the rest of the protein which provides the environment, which provides the conformational constraints, which makes the free energy the minimum to form that alpha helix or to form that beta sheet. That's why the problem is difficult. It's not a simple problem. We are trying to replace the rest of the protein interface with few staples here and there, while, and this part I didn't talk about very much, while keeping the sites which are actually going to be the active sites intact without completely freezing the peptide. If you freeze a peptide, nothing moves. You put too many staples everywhere, it's not going to do anything. Okay, so it's a hard problem. It's not, but it's a problem, I think, given where computational biophysics is now in 2020, it's a very amenable target. You have to be smart, you look at it, but there are many things to look at. You can say, okay, if I put this distance constraint in my MD simulation, can the protein open up to take the substrate? These questions can easily be asked and answered within hours. So I think the time has come for going into this direction.
Okay. Yeah. So as expected, like we have a few questions on COVID-19. So one of them just I will put forward. Mohit Dr. Behera asks, so can the protein part of SARS-CoV-19 not be degraded by any external protein or any enzyme? Like probably the active site he meant.
Okay, maybe I'll rephrase the question. Tell me if I understood it correctly. You were talking about that somehow to attack the SARS virus, attack the spike protein, and degrade that part which it uses to enter the human cell. Yes. So to do any such chemistry, first, you need to be able to target it well. And this is the problem for everything, even for cancer, right? You need to target the right thing. You don't want to put a very reactive molecule in the body because it's like drinking bleach that some famous person, let's not name him, prescribed some time ago. So you can't do that because it will be very reactive. So you need to first home in onto your target and then take out those your bullets right there. And this spike binding peptide is one of the ways that people can home in. As I said, it's a group of David Baker in California who have just come out 2 weeks ago in Science. These little proteins, the mini proteins, which actually have that peptide stable in a helix. So they home in with picomolar affinity, much better than nature's affinity, onto the SARS, the spike protein. And then you have to think of chemical ways to add something to it which can disrupt, not just bind, but disrupt the protein. It is possible, I think, but one has to be really clever about it.
So coming back to the stapling technology, so Saptak asked whether it can be used as a tool in treating Alzheimer's disease.
Okay, we are thinking of many proteins that can go in and do something in Alzheimer's disease. One of the ideas, I didn't want to talk about it because it is something that we're just working on, is to have, again, Alzheimer's protein decoys, which would be sensitive to something else, say pH or light even, and go in and integrate itself in the assembly of other amyloid beta, but this one is a synthetic one supplied from outside, and then you put a switch and it opens up because it has a little, for example, for the light-based one, which we're just having fun with azobenzene in it, and there is a certain distance that has to be maintained between two strands of the amyloid beta. You put UV light on azobenzene and it goes from cis to trans, and that distance opens up. And the hope is if it's part of the big assembly, that assembly will fall apart. So this is just an idea I'm talking about. I haven't shown it. So, but yes, it is possible that if you do it right and leave in a little place where your interruption can go in, where a bit of light or bit of whatever other molecules can go in, it can use that as a key, like a Trojan horse, and open up the gates. It is certainly possible, but nobody has shown it yet.
Okay. So probably the last question of this session. So Neova Paul asks, so do viruses or bacteria use such molecular protein staples to alter or change the structure of infected cells?
Okay. The stapling that I talked about is mostly synthetic chemistry stapling done outside of the cell, can be done inside of the cell by putting in some chemicals. The viruses do not use such things. See, the virus is just a message. Okay, it's just a message. It says reproduce me, and it uses all the machineries of our own cells to reproduce it. It's just, what is 29 kilobase pairs that the SARS-CoV-2 is. It's just the ability to put it in and use the cell's machinery. So it's not going to change the cell altogether. It's just telling that here is a piece of DNA, just reproduce it and pack it well. That's it. And the body obeys it, and our cells obey it. So it's not the strategy that viruses take. But we are talking about a strategy where we can, even in a living cell, we can do stapling. But then you have to do something first outside, put that molecule in, and then, maybe shine some light or something, and so that stapling happens.
Okay. Any more questions? Like if somebody wants to ask directly, you can raise your hand. So I can see two raised hands. Rahul Kumar, do you want to speak up? Okay, I think might be.
Soumik, can I ask Sudipta one question, please?
Yeah, sure.
Sudipta, do you think it's possible to do some kind of gene therapy by encoding these small peptides into the genome itself and having it co-expressed along with the proteins at some point in time. But is there a possibility, do you think?
Absolutely. Our next step is to actually encode this little gene inside a cell for the xenonucleus and express it, and to see if the ubiquitin functionality can be changed. Because when you actually have a gene put in, there is usually also you have a switch from the outside, IPTG or whatever. You can try to express it.
Yeah.
As it expresses it at the right time, can I change the folding of a particular protein? That would be the target. That would be the dream. Yes.
Okay.
Okay. So I think we have covered most of the question, but there is one question still. So why, Mohit asks, why is there no vaccine for the COVID yet? So so very question.
You
So,
read the newspaper, I read the newspaper, the vaccines are coming. And let's hope if one comes and one is not, one is effective before long, because our normal lives will have to depend on it.
Okay, so I think we have covered most of the important questions asked here. I would like to thank everyone for joining us today, and especially Professor Maiti. Thank you again for for giving coming here and giving such a stimulating talk. And we really hope that we might be able to host you in the near future physically in our institute once this COVID situation subsides. So thank you again. Thank you, Professor Maiti.
You're most welcome, and thank you very much for having me deliver this seminar. And yes, I would like to get there physically. It's just getting too boring, stuck in the thing. Okay, bye then.
Yes. So before all the participants leave, so since this is a seminar series and we will have talks every Thursday. So every Thursday you can probably visit our webinar website and you can find the link to join us here. And so our next talk will be delivered by Professor Amitava Patra, the director of INST Mohali on 1st October. So the link will be available on our website, and the students are requested to register if they want to avail for certificates. So we have planned to provide special certificates to students who will attend at least 10 talks of this series in a year. So we hope to see you all soon, and have a great evening. Bye-bye.
Bye.
Bye. Bye.